Okay, so in this video, we're looking at what kind of factors affect that rate of reaction. So we know that enzymes reduce the activation energy required, but what else is going to impact on how fast we go from reactant to product or substrate to product? So the first one we're going to look at is temperature. So you remember that we spoke about in the last video that all molecules have kinetic energy and therefore they're moving around. Well, what's going to happen if we heat that temperature up? They're going to get more kinetic energy and therefore they're going to move around faster. If we, say, took your classroom, hopefully you have a classroom, if not, play along. If you're in a classroom and there's, one student, okay, two students, and we want them to collide. Well, how often do you think they would collide if they were walking around really slowly? Probably less than if they were sprinting around. So imagine sprinting, colliding with a wall, coming off at a 90-degree angle, sprint, 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 sprint. The collisions are going to be more. Think about that with, say, 1,000 molecules, or let's say even 30 students in the classroom. Then if we increase the movement speed, then they're going to increase the number of collisions. If we increase the number of collisions, we increase the probability of that reaction to occur, especially if we're using enzymes. So, great, we want reactions to go fast, we just pump the temperature up. Anyone problems with that? Yes, a few of you there, I saw you thinking, well, hang on, enzymes have a specific three-dimensional shape that's held together by bonds themselves. If we increase the temperature too much, those bonds are going to vibrate too much to contain that bond and they're going to break apart, ruin the three-dimensional structure, and therefore they're not going to catalyze that reaction anymore, and you would be right. So that process is called denaturing. So denaturing is where we heat up, let's say we have this one here. If we heat that particular enzyme up too much, then those bonds start to break and we start to maybe change the shape of the active site, and now it's no longer complementary to our substrate or our reactant. So that's bad. So on the right here, I've got a graph, and this is a good way to sort of test your understanding of whether you actually understand the outcome of a variable on some factors, okay? So in this case, our variable is temperature, and we want to see how is it going to impact on the max products per second, okay? So I want you to pause this video in a second and draw a quick little graph and go, all right, if I increase the temperature, what's going to happen to the number of products per second? So if we did different temperatures, 10, 20, 30, 40, 50, 60 degrees, and measured a reaction, how many products would we get per second? And then bear in mind that we've got this problem of denaturing. So here's your chance and pause. Okay, we're back. Now, if you did it like me, we would be increasing our temperature, rate of products would be going up, going up, going up, going up until we reach an optimum. And then we're starting to break the bonds of our three-dimensional enzymes, and therefore they're going to change shape, no longer complementary to our substrate, and therefore the products aren't going to be made, and therefore we're going to drop off. And we're going to get to a stage eventually where we're going to get to zero, okay? It can't make any products. Now the reason why it doesn't, in my graph, well, it's a bit shaky, doesn't just drop off straight to here is because that temperature is going to affect enzymes differently. It's not going to be like a uniform, every enzyme's denatured at once. Some are going to be partially denatured, some are going to be fully denatured, some based on their positioning might not be completely denatured, and therefore we're going to get a bit of a curve. Now because this temperature is in degrees Celsius, let's say this is 0, we're still going to get possibly even some reactions to occur because they're going to have some kinetic energy, because that's not 0 Kelvin. So we're going to increase up to a certain point, and then we're going to start denaturing and we're going to drop off. So this point here is going to be our optimum. So whatever value this represents is going to be our optimum temperature for that particular enzyme. So that's temperature, okay? Increased temperature, more collisions, increased probability of effective reaction, and go too far, and we denature the enzyme. So if you're not using an enzyme, just pump the temperature right up. Okay, pH. Let's have a think about pH. Go back to the original videos. We're thinking about how do we even make proteins, enzymes, R groups, ionic, salt bridges, ionic bonding, therefore they're going to be affected by acid-base solutions. So our enzyme is going to have a specific pH that it's going to work best at, that's going to maintain the integrity of those ionic bonds, those salt bridges that we spoke about. So therefore, here's your chance. Pause the video, draw a graph of how you think the max products per second might vary depending on a varied pH. And pause. Okay, here's mine. So I'm going to grab one particular enzyme, and I'm going to say it's going to have an optimal pH, and it's going to be right here. And then either side of that, we're going to start disrupting the bonding of those R groups and things within the amino acids. So the secondary and tertiary structures of the protein, particularly the tertiary structure, and therefore it's going to change shape, we change structure, we change function, and therefore as more and more of those enzymes start to not work efficiently, we're going to drop off our max products. And so we get this sort of uniform look either side of our optimal pH. But we might be looking at an enzyme now in the stomach, and that's got a pH of 2, so those enzymes have evolved to work optimally at that pH. So it might look like that, where there's our optimum. So each enzyme is going to have this specific pH requirement, or this level that's optimal, and then it's going to have, 80% of the enzymes, 80% of the output of the enzyme is going to be working within like 1 or 2 pH levels or whatever, and it's going to be all different. So if you were a scientist, then you would then look into a specific enzyme and look at and do a pH experiment and look at how the pH affects that particular enzyme and work out its working range. Okay, substrate or enzyme concentration. So basically, let's look at enzyme concentration first. If we have an enzyme that can do, here's our enzyme, same one as before. If that enzyme can produce 1 product per second, okay, in 5 seconds we've produced 5 products. Potentially, if we can get them like in, out perfectly, we can produce 5 products in 5 seconds. Well, what happens if we want 1 million products in 1 second? How can we achieve that? We just have 1 million enzymes, okay? So as we increase the enzyme concentration, there's more active sites for the substrate to go into, or the reactants, to form products. So as we increase the as we increase the enzyme concentration, you'd expect the rate of product per second to increase. And this is the same for the substrate. If we've got a set level of enzyme, then as we increase the number of substrates, we're going to get more products per second, okay? Because more of those enzymes are now going to be occupied at any one time, okay? So have a think about that. Is there any limitations to that? Here's your chance to draw a graph, and pause, and go. Back. All right, so for both of these, we're going to expect to see an increase and then a leveling out, okay? Why a leveling out? Well, let's do enzyme concentration for a second. If we have a certain number of substrates, okay, as we increase the number of enzymes, eventually we're going to have more enzymes than what we have substrates to fill, and therefore we're going to get like a max product per second, but it doesn't matter how many enzymes we increase. Say we don't have enough substrate to fill those, it's going to... The same for substrates. Eventually, we'll increase the number of substrates until we're maxing out the number of available enzymes. So there's just not any enzymes available to put substrates into until they're clear. So that's going to make a rate that's going to then plateau. So hopefully that makes sense. If you're a little bit confused with those ones and want me to go into more detail, just pop a comment below and we can hit those. The final one, inhibitors. What happens if we start increasing the concentration of inhibitors? Remember, these are things that are going to block the active site and probably occupy the active site and not leave very often because they're not broken down. So have a think about what the graph might look like for those. And pause. Okay, this is what we'd expect to see. We're going to have this high level when the concentration is low, and then we're going to decrease. Okay, something like that, depending on the specific kinetics, and it's really more physics, you might get it looking like that, or quite linear, but you're basically going to get this decrease in product per second as we increase the number of inhibitors. Now, if you think you know which one it is out of A, B, or C, I'd love to hear your answer below with a justification. It could be those, those physics people. So jump in, let us know what you think it is, and I'd love to hear your answer. As always, if you like the video, give it a thumbs up, subscribe. Lots of videos coming out all the time, and we're going to be going into cell biology, organisms as a whole, ecosystems. So lots of videos to look out for, and hopefully this is helping you with your schooling. Mr. Dundon,